This lesson is about translation. So we finished off transcription. We've got a DNA template that has been used to create an mRNA strand, and the mRNA strand has moved out of the nucleus. In our cytoplasm, we have ribosomes, and ribosomes are going to help us make proteins. They are, ironically, proteins themselves, so those big folded chains of amino acids, which are going to help us make another big chain of folded amino acids out of our information that we've taken as a photocopy from the DNA. Now, the ribosomes are going to change our nucleotide sequence in our mRNA into a corresponding amino acid sequence. Now, if we have a look at this scrappy old table here, what this is an mRNA table. Now, we read, or the ribosomes read the mRNA in 3 bases at a time. So in this case, CCU, if that's on the mRNA strand, it will read it as the amino acid proline. There are quite a number of different amino acids, and lots of different things can code for the same one, but you'll also find that there are some individual ones and some other ones here. This is a start codon, so the mRNA strand should always start with AUG. The example we're going to give today doesn't, but you'll get the idea. To stop the protein being made, to stop more amino acids coming in and joining the big chain, you have UAA, UAG, or UGA. So a stop codon means, yep, we're done, stop collecting amino acids. The ribosome is quite intelligent and it knows these things. So as it reads the 3 base pairs, which we will also be calling a codon, it can draw in a piece of tRNA. Now, a piece of tRNA is considered to be transport RNA because it is a long folded piece of RNA. So remember, it's actually pointing outwards. The bases are on the outside of that RNA. And what it does is it exposes 3 of them. And those 3 are going to be the ones that match the mRNA. There are about 1,000 leaf blowers outside the door, so we're just going to have to live with it. Okay, instead of talking about it, we're going to give you a little bit of a feel for what this is. So, as ridiculous as it looks, this is a happy little ribosome, and that ribosome is going to read our mRNA strand 3 bases at a time. Okay, so the mRNA is going to have this here, and our first codon there is AAC. Now, the corresponding tRNA that will have those 3 bases exposed obviously will have to be complementary to AAC. So we will obviously be looking for a piece of tRNA that can expose UUG. And whatever is attached to the UUG will bring with it this one, which is asparagine, so ASN. Now the ribosome said, all right, we've got the correct tRNA here. It's brought its amino acid with it. So what we're going to do is we're going to keep feeding through and we're going to keep reading the next one. So GUA will correspond with a piece of tRNA that looks like this. So first one moves out of the way and we will start having a bond form between these 2 amino acids. And the chain will continue and continue and continue for as long as it needs to go. And the mRNA strand will continue to be read by the ribosome until we reach a UGA or another kind of stop codon. So this is a stop codon saying that it's going to pretty much end. So things we need to know. The ribosome is in charge of stringing together all of the amino acids here that are attached to the tRNAs, which are corresponding to the information being read on the messenger RNA strand. Okay, the free-floating amino acids are really attached to a big strand of tRNA, so those 3 bases are exposed at the bottom. So the tRNA is kind of swooping in, delivering with it a big amino acid that it is attached to. The way in which we know what amino acid is going to be brought is by reading our mRNA base and see what corresponding amino acid is going to bring it with you. Okay, so in that one, we can see this table on page 161 of our Biozone, or you can find it in so many other places. What we need to know now is that chain, that really simple chain of amino acids that keeps on building and building and building, is our protein. Okay, once some folding happens, and remember our proteins have multiple kinds of structure. It's got the initial amino acid sequence, then it folds over itself, and it can fold over other different proteins as well to form subunits. There's a lot going on there, but I'd recommend you watch an animation as well to help you out.